Hi, I'm Sheng. I'm a graduate student at Dr. Ed Monuki's lab at the University of California, Irvine. I'll be showing you how to extract RNA from neuroprecursor cells using this Bio-Rad total RNA mini kit. I've been using this kit for a couple years already at this lab, and it's always been giving me consistent results at high yields. So the cells that we use are E12.5 neuroprecursor cells that have been acutely dissociated from embryonic cortices. And we plate them usually in a 24-well tissue culture treated plate that has been PDL and laminin coated. And I plate them at 100,000 cells per well. Before I start, I have to tell you that you should work on a designated bench where you do your RNA work. First things first is to make sure your RNA bench is clean. Appropriate way to clean the bench is to use this RNA zap. Basically, it kills off any RNases. Reason being that the RNases basically destroys your RNAs. Other thing is to wear gloves always when you're handling the extraction, because your skin's filled with these RNases, so it's a big no-no. Okay, so what I have here is the Bio-Rad Total RNA Mini Kit. I've taken out its contents, so let me show you what they are. First thing is we have this 1X DepC treated PBS, and we have 70% DepC treated ethanol. We have total RNA licensed solution that has beta-methacryl ethanol added to it. Second is a low stringency wash that has